Hey, I'd like to welcome everyone this afternoon, and I know more people will continue to sign on, but I thought I would go ahead. I have the pleasure of introducing our speaker today, Dr. Isabelle Boileau. Dr. Boileau is a senior scientist at the Campbell Family Mental Health Research Institute at the Center for Addiction and Mental Health, CAMH, and the head of the Addiction Imaging Research Group at CAMH Brain Health Imaging Center. Dr. Boileau has established a world-class program of research at CAMH that has uniquely contributed to the international body of knowledge in the areas of substance abuse disorders, addiction, and comorbid conditions. She's also an associate professor in the Department of Psychiatry at the University of Toronto, and her work is funded by CIRH, NIH, Ontario Mental Health Foundation, and the Canadian Institute for Military and Veteran Health Research. Dr. Boileau completed her undergraduate work in psychology at Concordia University in Montreal and a master's at the University of Montreal before doing her doctoral work at McGill and a postdoc at Columbia before coming to the CAMH. I'm a longtime fan of Dr. Boileau, going back to her days at McGill when she was at that time becoming Dr. Boileau and doing really creative advanced work with PET and dopaminergic tracers right out of the gate. Now, many of you on this call know how challenging it can be to do PET. You finally get the human participant on hand, the perfect one, the eligible one, but the tracer yield may not be there on that day. Or the tracer yield is great that week, but the eligible participants are nowhere to be found. Isabelle has made this look easy with a string of early hits. With a PET team and a terrific group, she was able to lead studies showing dopamine release during placebos associated with drug administration, providing clear evidence for the kind of condition effects that have been demonstrated in preclinical research and subsequently in humans. She linked stimulant increases in dopamine to drug wanting and novelty seeking and showed that alcohol released dopamine in nucleus accumbens. All of this was even before she turned her attention to parsing the role of D2 versus D3 receptors, a topic close to many of our hearts, showing that a heightened response to amphetamine and methamphetamine users with the D3 tracer PHNO, and heightened D3 receptor availability in cocaine patients, in contrast to the usual finding of lower D2 receptors in drug-using populations. In recent years, Isabelle has turned her laser focus to the field of endocannabinoids, and the early news is that her contributions in this area will be every bit as impressive as those in her earlier work. We're in for a treat today as we hear about this newer work and the advances enabled by her creative use of translational neuroimaging tools, PET and MRI, to advance our understanding and treatment of human neuropsychiatric conditions. Dr. Boileau, welcome.
Thank you. What a nice introduction. I wasn't expecting that. I'm like, this is great. Excellent. And yeah, thanks for obviously the opportunity to present this work during this super isolating time. I think it feels good for me to imagine people listening to me talking other than, the dust in my room. So let me get started. Unfortunately, I'm not presenting lots of work on the dopamine system, but I did leave a little piece on that. If you're very interested in the dopamine system, you can still ask questions, and I'll try my best to tackle those. All right, so let me dive into this. So the focus of my talk is on the endocannabinoid system. The title is, Can Molecular Imaging Help Move Forward Endocannabinoid-Focused Treatment for Psychiatric Illness? So the focus of my lab and my work has been to use brain imaging to get a better understanding of molecular system in brains, druggable molecular system in brains, so that the systems that may be involved in mental health disorder. In the recent years, I've become interested in endocannabinoids, which are cannabis-like substances made in our body. And the question that I'm asking is whether endocannabinoid status is abnormal in some psychiatric conditions and whether endocannabinoid therapy could be useful. So let me first start by
Isabelle,
yes?
Isabelle, it seems that Chris is saying that he can't see your screen. I can see your screen, so
okay.
Let's troubleshoot for just a moment. Chris, I can see Isabelle's screen, but some people cannot.
Well, if it's just me, it's fine.
So can other people?
I can see it. Yeah, I can see it too. It's fine.
Okay, yeah. We have several people, so it's just Chris that's being left out. Sorry, Chris.
Hey, Chris.
Maybe you could sign in again. That's a little joke. Okay.
I'll do. Okay.
He's our AV person.
Thanks.
Aha.
Thanks, Isabelle. Please go ahead.
Okay, sure. So I was going to start introducing the endocannabinoid system for some of you that may not be familiar with it. So here's the obligatory cartoon of a synapse with endocannabinoid components. So as I mentioned, endocannabinoids are cannabis-like molecules that are produced abundantly in the brain. And the two main endocannabinoids are anandamide and 2-AG. And those are made from membrane phospholipids. And what's interesting about this system is that they're released on demand, and they travel from the postsynaptic neuron upon their release to the presynaptic neuron. And this form of neurotransmission is called retrograde transmission. And in the presynaptic neuron, they act at cannabinoid receptor. The most known is cannabinoid type 1 receptor, where they would quiet down the activity of the presynaptic neuron. So it's believed that the endocannabinoid system is really one of those systems in brains that modulates or that moderates activity within important neural circuitry, including the mesolimbic dopamine system. And one that's also been studied a lot is the amygdala-related circuitry involved in stress and emotion processing. Once the endocannabinoids are released, and they're eventually degraded by two key enzymes. The one that I'm interested in is fatty acid amide hydrolase, which degrades anandamide, and monoacylglycerol lipase, which catabolizes 2-AG. So what happens when either 2-AG or fatty acid amide hydrolase don't function well is that you end up with this accumulation of cannabis-like substances in the brain. Which brings me to talk about Jo Cameron. So maybe some of you have heard of Jo Cameron, have read about her. She is a Scottish woman, a mother, and used to be a teacher, but she became known to the scientific community as someone who doesn't feel pain. So she's insensitive to the heat of chili peppers, and she doesn't know the extremes of rage, anxiety, or fear. And her condition is believed to be attributable to, let me see if I can make my, attributable to two genetic mutations. One is a mutation in a pseudogene called FA-out. It's a new pseudogene, FA-out. And the other is a common polymorphism that affects the FA gene itself. Altogether, those two genetic quirks have led to a condition in which there's very low levels of Fa, and as a consequence, higher levels of these endocannabinoids, particularly anandamide. So this is the subject here, Jo Cameron, relative to wild-type individuals. So, and it's believed, of course, that this increase in anandamide, probably in brain, is what is responsible for this, happy-go-lucky demeanor that she has. So with a case like Jo Cameron, it's not hard to see why modulating Fa is perceived by the scientific community as having a huge therapeutic potential, particularly in reducing pain and anxiety. And in the last decade, development of endocannabinoid enzyme inhibitors, for example, MAGL or fatty acid amide hydrolase inhibitors, have been avidly pursued, inspired, of course, by the medicinal properties of cannabis. Here, the main idea is that if you inhibit these enzymes and you increase naturally the levels of endocannabinoid, this may have less of a side effect or an adverse effect than activating CB1 receptors directly through THC. And this, of course, has huge potential for as anti-inflammation, analgesia, anti-anxiety, anti-depression. So currently, although the development of these endocannabinoid enzyme inhibitor has been pursued, there are no FA or MAGL inhibitors that are approved for any indications in humans. And bottom line is, I believe that a better understanding of the endocannabinoid system in the living human brain can help us, move forward some of these drugs that have been developed. So as you know, the failure rate and the cost associated with developing drugs for the central nervous system disease is very high. In all disease area, though, there are several common requirements for designing successful new treatment. For one, obviously, the disease condition must be well understood and a target must be identified. Once we have that, then we can start developing programs that move forward, that develop molecules that have desirable qualities. These eventually get pushed through IND enabling processes and through clinical trials. But recently, PET imaging has been helping providing data to support key decision making during the drug development process. And this, obviously would be to reduce the risk and the cost of later failure. So because PET is the only way to investigate molecular system in humans in vivo, it can provide proof of concept and help detect and measure the expression of these targets that we're trying to modulate. It can also track drug engagement with the target and establish early evidence of treatment response. So how do we look at the endocannabinoid system in the living human brain? So with PET, obviously. So our institution has developed, let me just pull this up here, has developed a probe for fatty acid amide hydrolase. So this is the molecule here. So it's a prototypic irreversible Fa inhibitor, ERB691, that's been labeled with carbon 11. And this radio tracer, we've had a lot of fun with it. It's a good radio tracer. It does what it's supposed to do. So here on the right is a panel showing curve distribution in the brain. And this tells you that our tracer distributes in the brain where you'd expect to see Fa. We also shown that it's selective and specific for the enzyme. We conducted a study using a Fa inhibitor, and what you can see here is almost a complete loss of the curb signal in the brain after a clinical dose of this Fa inhibitor. We've also shown that it has very good retest reliability. So here are time activity curves, so standard uptake values for test and retest showing almost complete overlap. So this is in healthy controls coming in 1 month apart. So excellent test-retest reliability. And we've also shown that the behavior of the tracer could be modeled using a two-tissue compartment model for irreversible trapping in which lambda K3, so when I talk about lambda K3, I'm talking about our main parameter of interest, that is an index of Fa level in the human brain. Right. Okay, so let me move forward here. So over the last few years, the focus of my lab has been to use brain PET imaging of Fa with our radio tracer curve to gain a better understanding of whether endocannabinoid metabolism in human relates to psychiatric illnesses. And I'll present some work in substance use disorder, because I know you're interested in addiction and substance use disorders, but I'm also bringing in some pieces of work from our work in people with post-traumatic stress disorder, as well as social anxiety disorder. Another line of research also has been to try to relate Fa levels in brain with risk phenotypes using multimodal imaging approach. I will show you some of that work as well. Some of what we know about the role of Fa in human has come from our studies of humans who have a common loss of function Fa mutation, which is one of the genetic quirks that Joe Cameron inherited. So this Fa SNP occurs in about 40% of Caucasian, and it involves the conversion of a cytosine to an adenosine at position 385 in the Fa gene, which results in a switch in the amino acid sequence at position 129 from a proline to a threonine. And what ends up happening is that there is an enzyme that is easily degraded or unstable, and so very low levels of Fa activity and Fa protein in the brain. So we've indeed shown using our PET radioligand curve that individuals with 1 or 2 copies of the minor allele of this polymorphism have lower levels of Fa in brain, at 26% lower Fa levels or curb binding in brain. So let me just walk you through this graph because I use a lot of these graphs in my presentation of data. So what you have here on the x-axis are brain regions of interest, and typically we use a template that covers the whole brain. So you have all the brain regions of interest here. And on the y-axis, you have our parameter of interest, which is curb lambda K3 across all the brain. The empty circles are individuals with 1 copy of the minor allele, and the wild type are in gray. The black dot at the bottom is the one individual in our cohort that had 2 copies of the minor allele, so very low levels of Fa. So what you can see here is across the whole brain, there's very lower levels of curb binding Fa in brain of individuals with this polymorphism. So, again, this is the first evidence that this SNP affecting Fa is actually functional in the living human brain. And here I really want you to appreciate, the rarity of these findings in which a single locus in the genome is associated with a measurable molecular change in brain. So there's not a lot of studies actually that have been able to do this. All right, so, but, how do individuals with this polymorphism behave? So to keep it simple, maybe even simplistic, on one hand, it's been described that individuals with the C385 variant, so the ones that have lower Fa, higher levels of these cannabis substances in brain, have increased approach behavior, such as increased trait impulsivity. They also have a tendency for greater drug and alcohol use and obesity. They have greater ventral striatum response to reward, higher rates of substance use disorder, particularly of sedative drugs, and also higher rates of problematic drinking. And on the other hand, they have traits that, I guess are kind of like Joe Cameron. So they have lower trait anxiety, lower amygdala response to threat, a higher functional, so greater functional connectivity in the sphere-related circuitry. They tend to feel less the effects, the negative effects of withdrawal from cannabis and from heavy drinking. And we have some data also that complement these findings that that already exist. And for the most part, the preclinical literature has been in line with the human genetic studies. So mice with genetically impaired Fa, so these are either Fa knockouts or Fa knock-ins. So these mice have been engineered to replicate the C385A human SNP. As well as pharmacological manipulation. So if you give Fa inhibitor to animals, and this I guess has been really important in addressing here whether the phenotype seen in behavioral genetic studies is directly caused by elevated Fa or is just the result of neurodevelopmental effects. So these mice, they tend to show impairments. They show a range of behaviors that are similar to what we've seen in humans. So decreased fear avoidance, increased pain threshold. They also have greater functional connectivity in the fear-related circuit. They tend to drink more, but feel less the sedative or the effects of alcohol. So because of the relevance of Fa in anxiety and stress-related conditions, one of our objectives was to ask whether Fa is elevated in fear-related conditions and whether Fa was related to function within neurocircuitry involved in fear and emotion processing. So this is a study that we've just published in the Journal of Neuroscience and Psychiatry. So in this study, what we did is we recruited 31 healthy controls who participated in both our PET imaging scans with curb that measures Fa in brain, as well as they did the resting state fMRI scan, in which we investigated the connectivity between the amygdala and prefrontal cortex and DACC. So what we found in this study, and you can see this here, is that individuals with lower Fa in brain, so this is on the x-axis here, have higher strength of connectivity between the amygdala and brain regions, including the subgenual ventromedial prefrontal cortex, as well as the DACC. So both regions involved in fear and emotion processing. They have different roles, but they're both involved in the process. So in both cases here, a negative relationship, meaning the lower the Fa, the higher of these cannabis-like substances in brain, the greater the strength of connectivity. So, and this is what's shown there. So, well, the finding is interesting as it suggests, in line with the preclinical literature and with earlier neuroimaging studies investigating the link between resting state connectivity and this Fa mutation, that brain levels of Fa affect amygdala circuitry involved in fear and emotion processing, and as such, drugs that inhibit Fa may regulate this circuitry, known to be abnormally functioning in people with post-traumatic stress disorder, anxiety, and mood disorders. I got distracted. I heard my kids coming in. Ooh. All right. So along those lines, we've also shown in an overlapping sample of 28 healthy controls that bold reactivity to a perceptual phase task. So this is the perceptual phase task that we use. So people were seeing angry or threatening, or fearful faces, which engages the amygdala. And here we've shown is that the magnitude of this bold response to threat is related to Fa levels in the amygdala. Here the relationship is positive, so the greater the threat, the greater the Fa, so meaning the lower the levels of, let's say, endocannabinoid and anandamide in brain, the higher the response to threat. Once again, these results are in line with what could have been predicted from animal studies and suggest that Fa inhibitors may decrease amygdala response to threat, a response which we know through decades of work is abnormal in some psychiatric illnesses, including post-traumatic stress. So, these findings are particularly relevant to ongoing clinical trials of Fa inhibitors for fear-related conditions. So here is an example of this. So this is work from the group of Dr. Heilig, showing that if you give a clinical dose of a Fa inhibitor to healthy humans, that this decreases response. First of all, that increases anandamide, but it decreases fear learning, autonomic stress reactivity, stress-induced affective response, suggesting that Fa inhibitors may decrease response to fear and anxiogenic effects of stress in humans. So given the interest of pharmaceutical firms in developing Fa inhibitors for anxiety disorders and post-traumatic stress, we became interested in addressing in a set of simple proof of concept studies whether curb binding, or Fa, is elevated or upregulated in people with social anxiety and post-traumatic stress disorder. So what's on this slide is essentially the design of these cross-sectional studies in which we invite people with post-traumatic stress or people with social anxiety to come in and do our PET studies. So they typically have a very lengthy screening interview, and this is followed by an MRI and a PET, which we tend to do on the same day. The MRI is about an hour long, and the PET is about 90 minutes. And here are the results of some of these studies. So in this study that's still ongoing, unfortunately, we got shut down, as you probably did, in March, last March, and we haven't been able to, finalize the analysis or continue to recruit. But at the time of our departure from the lab, we had recruited 12 people with social anxiety disorders. So these are 12 people that don't have other comorbid conditions. They have social anxiety. Some of them have moderate mood disorder, but they don't have substance use disorder and they don't smoke cannabis. and they were matched with 30 healthy controls. And what you can see here, again, lambda K3 values for curve on the y-axis and brain regions of interest across the x-axis. And we found a slight increase, and we're starting to see trends for increased Pha in some brain areas in people with social anxiety disorder. Again, we need to obviously continue this study, and we're really excited to get things started again there. So these are some brain areas. And in other brain areas, these are cortical brain areas, the effect was less pronounced. So it may be, again, another question that we're wondering is whether those effects are region-specific or are they a brain. But what was interesting to me in that study was this nice relationship that we found between Pha levels in brain and scores on the Leibovitz test for social anxiety. So this is a test that measures anxiety and how fearful a person is in a situation and how much they have to avoid this situation because of their fear. So, which suggests again that at least some people with high levels of social anxiety have higher levels of Pha. So in contrast to the study in social anxiety disorder, in people with post-traumatic stress, so here we recruited 16 people with post-traumatic stress and 29 healthy controls. And again, here, sorry, the scattergram is a little bit small, but brain regions of interest here and curve lambda K3. And what you can see here is that unlike our study in social anxiety, here, the people with post-traumatic stress are in blue. We didn't find an elevation of Pha as we would have expected from animal studies. Instead, we found a trend for lower Pha in people with post-traumatic stress disorder. Again, the study is ongoing. We're not sure which way it's heading. But right now, it certainly doesn't look like people with post-traumatic stress have elevated Pha levels in brain. Sorry. Just a quick summary so far of the findings. So, what I've shown you is that our multimodal imaging data in healthy control are in line with models suggesting that upregulated Pha may contribute to abnormal frontal amygdala circuit function. These findings support use of Pha inhibitors in disorders in which abnormalities in these circuits are suspected, so post-traumatic stress and social anxiety. Our findings in social anxiety support the view that upregulated Pha may contribute to the pathology. And our findings in post-traumatic stress are not in line with imperfect animal models of PTSD in which differences in the timeline or acute state may explain discrepant findings. So here what I mean is that our finding in post-traumatic stress disorder is disappointing because you look at all the preclinical literature and you would have imagined that we would have found elevations in Pha. But understanding a little bit more the preclinical model, it's not surprising that we didn't find elevations in Pha. And maybe the study to do is to sort of more be in line with the preclinical literature is to look at Pha during an acute exposure to stressor. Something that I'm not sure we'll get into, but maybe. So a second line of questioning that we embarked on focused on addressing whether Pha is lower in substance use disorder. So here I'll remind you that both animal and some clinical studies suggest that greater drug use is associated with lower levels of Pha, higher levels of anandamide. So our general objective was therefore to test the hypothesis that substance use would be associated with lower Pha levels in brain. It's been proposed that Pha inhibitors could be used as a substitution therapy to reduce the negative effects of cannabis withdrawal. So this is a study that was published last year showing promising results, giving a Pha inhibitor to men with cannabis use disorder during cannabis withdrawal and noticing some reductions in craving. So what this was one of the first studies we did using our radio tracer curve is a study in cannabis user. And what we found here is that, this panel on the right shows the brain of a healthy control receiving the radio tracer curve, showing bright signal everywhere, and the brain of somebody with cannabis use disorder during very early abstinence from cannabis. So this is overnight abstinence, showing a reduction in Fa binding across the brain. And here what was interesting is that we also found a relationship between this binding in brain. So low Fa was related to higher cannabis metabolites, suggesting that more recent, heavier use of cannabis may be driving Fa levels down. So in light of the result of the clinical trial, one may think that decreased Fa in cannabis user may be a compensatory good thing, that reduces early symptoms of withdrawal. But we don't know. So I guess the important study asking whether Fa levels increase during acute withdrawal is being done now. So we're actually in the midst of this study, which was stopped by the pandemic, but maybe next time I'll be able to tell you a little bit more about that. Sorry. Okay, so one of the most studied area in substance use disorder research in Fa is alcohol use disorder. So here, preclinical literature has shown that alcohol exposure downregulates the enzyme, and that if you block the enzyme, either genetically, so Fa knockouts and Fa knock-in mice, or pharmacologically, that seems to increase drinking and also change the way the animal reacts to alcohol. So Fa knockouts, for example, appear to not as easily get drunk relative to wild type. And these preclinical findings are sort of in line with studies looking at what happens in individuals with the C385A polymorphism. So this is a study that was published recently showing that in individuals with alcohol use disorder, the minor allele of this polymorphism is associated with greater rates of drinking. And I'll show you some of our data on the same question. But if you consider what exists in terms of literature on Fa levels in the brain of a human, there really wasn't a lot of data there. So there's been that I know only 2 post-mortem brain studies looking at Fa levels in brain of people who have alcohol use disorder, and the 2 studies appear to be contradictory, in which one study finds Fa to be lower and the other study doesn't find anything. So, when we started our studies looking at Fa in people with alcohol use disorder, I think, there really was a gap in our understanding of the status of Fa in the brain of humans. So this is the first study we did in people with alcohol use disorder. It was published at the beginning of this year in the Journal of Neuropsychopharmacology. We scanned 14 people with alcohol use disorder and 25 healthy controls. The people with alcohol use disorder completed 2 PET scans, one during early abstinence from alcohol. So this is 3 or 5 days, just as long as they were stable, after alcohol use, and the other scan was done after 3 weeks of abstinence. And this again, curve lambda K3 here on the y-axis and brain regions of interest across the board. And what you can see here is that in individuals with alcohol use disorder, these are the triangles, the open triangles. Sorry, maybe it's a bit small. They have lower levels of Fa across all brains investigated, brain regions investigated. And interestingly, what we found is that the lower the Fa, the greater the self-reported alcohol drinks a week. So people that have lower Fa, higher levels of anandamide tend to drink more. So this is what happens during protracted abstinence. As I said there, we look at FA during early abstinence at the beginning, during the first week, and then 3 weeks later. Only 9 subjects managed to complete the second scan. And what it looks like is there's a tendency for normalization of FA levels as during protracted abstinence, so that FA levels may be transiently low during early abstinence and then recover. But the data, the scatter is quite big here during the second scan, and some people increase, some people don't change at all. So I guess the goal of the study that we're doing now is trying to really understand the trajectory of the changes in FA and see how it relates to behavior. Again, another study that was unfortunately stopped by the pandemic, but maybe I'll have more on that when I next talk to you. So in our studies, we also measure peripheral levels of endocannabinoids in all subjects that participate. And this is what you can see here. These are levels of anandamide, but also of OEA and DHEA. And these are healthy controls and people with alcohol use disorder during early and protracted abstinence. And I guess the main finding here is that relative to healthy control, individuals with alcohol use disorder during early abstinence have higher levels of anandamide, as well as higher levels of OEA and DHEA. And here we're not talking little differences, 76%, 60%, 32% increases in these FA substrates in peripheral blood. And interestingly, and this is, the only study in which we find a blood-brain relationship was in this study. We weren't able to find this in cannabis users, but we find this relationship between blood and brain. So this inverse relationship in which the lower the levels of FA in brain, the higher the levels of FA substrate. Another question that was of interest to us was to figure out whether we could predict relapse based on FA levels in brain. And even though this is a small pilot study, we tried to see whether there was a link between relapsers and FA levels in brain. And indeed, that's what we find here. So again, brain regions of interest, FA levels and curb levels. And in individuals that relapse, FA levels are lower across the board. And not only FA levels in brain are lower, but peripheral levels of FA substrates are higher in the blood. So that's true here. 12% higher levels of anandamide, but here DHEA, 96% higher DHEA levels in people that relapse. I mean, this is really interesting. It's a very small study, but I'd be interested in replicating these findings. What if DHEA could become a predictor of relapse? Anyways, one that we'll follow up as our studies build on. So animal studies have shown that rats that are bred for alcohol preference have lower FA expression and activity. So this is what's shown here. Even before they drink. So it's been hypothesized that impaired FA function may increase risk for later, increased alcohol intake and alcohol use disorder. So with that in mind, we decided to design a study looking at FA as it relates to risk for development of alcohol use disorder. We recruited in the study youth that are at the beginning of their drinking career, and they either had a family history of alcohol use disorder or they didn't. And the first question was, is there a difference in FA levels in brain in people that have a family history of alcohol use disorder? The answer to that is no. We didn't find that FA levels differed in people with a family history of alcohol use disorder. What we did find in this study that was interesting is these marginal, they're marginal, but they're still interesting, relationship between FA levels in brain and audit scores. So the audit score, as you may know, measures, pathological drinking. And so here, the ones that drink more and have problematic patterns of drinking have lower FA levels in brain across brain regions of interest. And this is interesting, even though it's marginal, it's in line with a study that's already been published in people with alcohol use disorder, but with our own studies here. So this study we're working on publishing now, it is a study, it's a large cohort study in youth who binge drink, and they were genotyped with this for this Fa polymorphism, and we show in this cohort that individuals with higher audit scores have this Fa polymorphism associated with lower levels of Fa. And this is true for total audit scores, but also for audit C, which is, I guess, the index of the amount of consumptions. This is another, I think, interesting finding that comes out of this study in youth who drink. We find in these individuals that Fa levels in brain is related to feeling less the negative effects of alcohol. So the lower the Fa, the less somebody feels the negative effects of drinking too much. So they don't feel the negative effects of a hangover. And they also feel less sedated from drinking. And there's a parallel to be made here between our finding and the preclinical literature. So heart rate variability is a marker of parasympathetic tone, vagal input, and low resting heart rate variability is one of the most commonly reported risk markers for alcohol use disorder. So in our study, we also looked at heart rate variability in youth during an alcohol infusion paradigm. And what we find here is that Fa levels in brain are associated with lower heart rate variability during alcohol infusion. Again, still, pilot studies, not a lot of cases, but if these results hold, it suggests that lower Fa levels in brain are associated with some of these known risk factors or phenotypes of risk for alcohol use disorder, including heart rate variability, low response to the negative effects of alcohol. So the summary of the results in substance use disorder, let me just pull them all out, it's easier. That. So Fa levels are lower in cannabis use disorder and related to heavier use, which could mask withdrawal symptoms. In alcohol use disorder, Fa are transiently lower and peripheral levels of anandamide higher in recently abstinent subject with alcohol use disorder compared to controls and relate to heavier recent alcohol use and relapse. We didn't find that Fa level is lower in people with family history, but we did find that low Fa is associated with greater rates of drinking and decreased self-reported negative effects of alcohol and lower heart rate variability during alcohol infusion. All right. So this is the last data that I'm going to be showing, and I put it in here because it has to do with dopamine. So this last study that I'll talk about, was, I guess, trying to address why it may be that low Fa is associated with higher rates of substance use disorder. And as mentioned earlier, I've been really interested in the dopamine system and its link to addiction. So because of the known importance of the dopamine system in addiction-relevant behavior, we wanted to know whether inherited differences in brain Fa levels, so people with the Fa polymorphism, is related to differences in dopamine receptor levels, D2 and D3, and particularly D3 because some of the findings from my earlier work suggest maybe the link between D3 and addiction-relevant phenotypes. So in a retrospective study, we genotyped 79 individuals who would have participated in our PET studies using a tracer PHNO. So PHNO, as you know, is able to maybe dissect D2 versus D3 component of binding, so that we used it for those reasons. And what we found was that, and that's illustrated here, is that PHNO binding across different brain regions. Some are D3, more D3 selective, but that individuals with one or two copies of the minor allele have higher D3 receptor, higher binding in areas that are more selective to D3. So that's in the ventral pallidum, in the globus pallidus, and in the limbic striatum. However, we didn't find it higher in the substantia nigra. So when I first saw these results, of course I didn't believe it. Who would? But so what we decided to do is to do a reverse translation study, and we borrowed or were offered mice with that have this C385A genetic polymorphism. So those are the knock-in mice. And we investigated PHNO binding in these mice, and we were able to replicate the finding in humans. So what you see here again are brain regions of interest in mice, in which mice with the C385A variant, the minor allele, have higher PHNO binding in the island of Calleja, ventral pallidum. So this is the area in the brain in which D3 in which PHNO binding is specific to D3. So we found that both protein levels and mRNA were elevated in the C385A knock-in mice. Again, I guess an image showing what I've just told you, increased mRNA in island of Calleja and of these knock-in mice. So, this study, I blew my mind. I really like it. I thought that results, it really never happens like that in the real world, does it? Okay, and so to conclude, so LOFA and increased endocannabinoid tone may be related to motivation to use drugs and increased tolerance to alcohol, for example. Whether LOFA is an acute compensatory response or an inherited or acquired biological vulnerability is not known. Obviously, the exact mechanism linking LOFA with more drinking or greater tolerance is not known, but may involve, GABA, decreased GABA, which is, a study that we could potentially plan on doing, or maybe it's related to the mesolimbic dopamine system. So more of these studies, I think, are going to be needed, in which we try to understand the link between the endocannabinoid system and its effect on other neurotransmitter system, dopamine, GABA, for example. Longitudinal studies comparing FA against clinical symptoms would help in understanding whether FA might represent a useful therapeutic target. So still a lot of work to do, and hopefully this pandemic ends soon so I can get back and answer some of the questions that I'm trying to. And last but not least, let me acknowledge obviously the very nice group of people that I work with and that I miss a lot during those times. So Alan Wilson here is the person who developed Curve for us. Dr. Sylvain Oro is the director of the Penn Center at the time where I was doing these experiments. And this is my lab, Laura Best, who does most of the work in alcohol use disorder, Ismail Mansouri working on the cannabis piece. Duncan Green is the one responsible for most of the multimodal imaging stuff that we do in the lab. Nadia Bocci, responsible for some of the cannabis work. Erin Godet does the stuff on post-traumatic stress disorder, and Mashal Hamed works on social anxiety. So, and of course, this would not be possible without some of the funding. So I'd like to acknowledge the funding agency. And that's...
Let's unmute so that we can clap for you, Isabelle. Let's unmute.
Thank you.
And I know you'll have questions. There's a lot of cannabinoid appreciators in the audience, so I'll let other people go first. I just wanted to quickly say, though, that polymorphism containing, that much control over the variants is just extraordinary. It is.
Yeah.
And so
it's a problem in the analysis, but it's quite.
Yeah, but it's a great tool, and so that's just stunning. But I'll shut up and let people say things.
Okay.
Cannabinoid appreciators or others, please ask Isabelle. Why?
Isabelle, I have a question about your metrics for measuring your cannabinoid uptake of your tracer. I noticed you're using lambda K3, which I know is supposed to be better bias and lower error for instead of using K3. But just out of curiosity, did you also look at the more traditional flux like Ki and K3? Just curious how those worked out for you.
Yeah, so we do, and if we want to do parametric images, unfortunately, so we use KI for parametric images. So what we've done, what we're in the midst of doing, is trying to create those parametric images of the data that I've just shown you. But overall, there are no differences in, it doesn't change the results. Like we've done it for the alcohol study. I don't think we've finished doing this for some of the other study, post-traumatic stress and social anxiety, but the results don't change if you use KI. And in fact, again, we need to use KI because that's what we use it to do voxel parametric maps. Yeah.
Okay. Just out of curiosity, did you try just the internal parameter, just K3? And was that too noisy? I'm just curious if it was.
No, I'm sorry. We haven't used just K3.
It wouldn't be recommended. I was just curious if it was truly as bad as rumored. Sometimes a good negative confirmation is interesting as well. Thank you.
Okay, thank you.
I just have a quick question, and other people will come up with theirs. I know that you've been looking at these different populations, and it's just intriguing. I wonder if there's an association between people who have this polymorphism, which actually, the one that you're talking about that affects FA, with other polymorphisms like FKBP5 that seems to be associated with an enhanced response to stress. So is there, basically a high cortisol, over-respond to stress? It's a favorite polymorphism that also seems to control quite a bit of variability in people that study anxiety, PTSD. And I just wondered if these things never go together, if they are something that would be, if you had the favorable polymorphism, you could be buffered. I didn't know whether you looked at any other associated polymorphisms.
We do collect, so we collect, and I know that Dr. Hamed Hariri has looked at CRF polymorphism as it relates to, because I think CRF is in the pipeline of downstream or upstream effects on the endocannabinoid system. So I think there are studies trying to look at the link between the FA polymorphism and other polymorphisms. I just don't know what the results are.
No, that's okay. I know these things are complicated. I just was curious. And the other one was sort of a clinical question. I mean, I know that there was a great deal of excitement and hope about a CB1 antagonist being potentially therapeutic. And, cannabinoids have been tricky in terms of these therapeutics, and you can begin to see why in terms of the animal data with if something reduces your hangover, then, a hangover people have talked about might be one of the things that would be something that would reduce your drinking. And so what are your thoughts about going forward for these things in substance use disorders? What do you think?
So FA inhibitors in substance use disorder, I've been asked this question a lot, given some of our findings that lowering FA may motivate people to drink, or they might feel less the negative effects of alcohol, then that's probably not a good thing.
That's sort of what I was asking.
That's probably not a good thing. And, the answer is it may be a question of timing. When does one give a FA inhibitor, or when does one, in what, so for example, somebody with post-traumatic stress disorder or with social anxiety disorder that may benefit from a FA inhibitor, should they be not prescribed FA inhibitors because of the possibility that this may trigger more drinking? Mm-hmm. And I don't know the answer to that, but I think it may be a question of timing. So when is the right window to give FA inhibitors to people with who also have comorbid substance use disorders?
It's challenging. It is challenging, especially since some of the substance use patients, have other comorbidities like anxiety or things where you might want to, right? And so it's kind of an interesting challenge in some ways to sort of figure to parse it. But the endocannabinoid system is so intriguing. The other question, really quickly, and then I promise to shut up and let other people, is about manipulations with this polymorphism with PET and with cannabidiol, with something that would actually be blocking effects. What are your thoughts? Have you looked at that? Are you looking at that?
Yeah, we had started to look at the effects of, pitched a trial of cannabidiol and social anxiety disorder. I mean, one of the studies that and I don't know, maybe some of you are already doing these studies to figure out what cannabidiol does on the endocannabinoid system. I mean, some suggest that cannabidiol could be acting as an inhibitor of FA, but I don't know that these results or these findings are robust, and whether it'd be worth it to do a trial looking at the effects of cannabidiol on FA inhibitors.
Yeah, it seems like it's a timely juxtaposition for someone who for someone who has the tools and that uses them as well as you, so that having an adequate sample size doesn't seem to be as much of a barrier. And even if we ever get out of the pandemic, you'll have a long list of things that you want to do and catch up on. But I'm just curious, because it seems that a lot of attention is being given to cannabidiol, and it would be interesting to know mechanistically what is going on and for whom.
Yeah, these studies are, again, we're all in the planning, and we're actually well advanced in the planning. One of the things that happened in 2018 is that cannabis became legal in Canada, and with that, it became impossible to do studies. It really became a nightmare. It's like our regulatory agencies still haven't figured out how we can do this, and so
That's interesting.
It's ridiculous.
Well, so what are the barriers that you encounter?
The barriers are complicated. I mean, it basically takes, obviously, an investigator's brochure, some information from a cannabis provider, and these companies are unable to provide those somehow because it's too much resources on their end. So basically, it comes down to money. The people that are selling cannabis, they're not in the market of spending money for research.
The research part.
Yeah.
That's unfortunate, because I think that's something that would really be in advance. But I will be quiet. I know there are other people probably who might have a question or two, and we have a minute or so left. How expensive is your tracer to make?
Well, we are being charged $3,800 per scan, which is not that much Canadian dollars. So, so per scan, per arterial scan.
Okay. Yeah. Just curious. I know that all of them are expensive, but I just wondered.
They're all the same. I mean, arterial scans are more expensive to run than non-arterial scans, and that's
right. Absolutely. Other people have questions for preclinical or clinical thoughts?
Yeah, and I just said thank you so much for bringing. Oh,
I think it's Corinne. Corinne, you're not. I'm not hearing you. Can you hear the question, Isabelle?
Nope.
Corinne, you might try turning off your video and just have your audio improve. Oh, looks like we lost her.
Uh-oh. Sorry, I know Corinne probably had a question, but if she did, hopefully she can follow on with Isabelle. Are there any other questions? We are at 4:00. Okay. If not, Isabelle, thank you so very, very much.